Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 5, Fossils, Dating, and Ancient Environments. In this module, we explore how fossils form, how we date them, and how we reconstruct the ecosystems of the past. These are the tools that allow us to piece together life's deep history, our only way of seeing into the millions of years that came before us. In this section, we investigate ancient climates, oxygen and carbon isotope evidence, and how environmental changes influenced evolution. If the previous section helped us place fossils in time, this one helps us imagine the worlds those organisms actually lived in. Let's revisit why reconstructing paleoenvironments is essential for understanding evolution. Fossils give us anatomy, but environments give us context, the pressures and opportunities that shaped how species lived, competed, and adapted. If we want to understand why certain traits emerged when they did, we have to understand the climates and ecosystems that surrounded those early organisms. Think about this as setting the stage before introducing the characters of an evolutionary story. Let's look at how scientists begin this process. One of the most valuable tools comes from tiny marine organisms called foraminifera, whose shells record oxygen isotope ratios. When these shells accumulate in ocean sediments, they form a continuous climate archive stretching back millions of years. By studying these isotopes, researchers can estimate global temperatures and identify major climatic shifts, periods of warming, cooling, or rapid fluctuation. These shifts didn't just influence ocean temperatures, they shaped rainfall patterns, vegetation zones, and ultimately the habitats available to our primate and hominin ancestors. On land, sediments offer their own environmental clues. Grain size, soil chemistry, and the presence of certain minerals can tell us whether an area was once a lake margin, a woodland, or an open grassland. Think about how different life would be in a dense forest compared to a savanna. Each landscape presents challenges and opportunities that leave traces in the geological record. A layer rich in volcanic ash, for instance, might indicate rapid burial and excellent preservation conditions, while a layer dominated by fine silt could point to slow accumulation in a floodplain. You might wonder how we reconstruct vegetation without preserved plants. This is where carbon isotopes become useful. Different types of plants, C3 trees and shrubs versus C4 tropical grasses, have distinct carbon signatures. When animals eat these plants, those signatures become encoded in their bones and teeth. By measuring carbon isotopes in fossil enamel, we can infer what kinds of plants dominated the environment and what kinds of diets ancient mammals relied on. A shift toward more C4 signatures, for example, indicates expanding grasslands, which played a major role in shaping the evolution of early hominins. Let's look at the bigger climate patterns. Throughout the Miocene, Earth experienced gradual cooling that altered ecosystems across Africa and Eurasia. Later, during the Pleistocene, repeated glacial cycles created alternating periods of wet and dry conditions. These fluctuations didn't just change landscapes, they changed evolutionary trajectories. Lineages diversified or went extinct depending on how well they adapted to these changing conditions. Understanding these patterns allows us to interpret why certain primates thrived during one epoch but struggled in another. As we put all these clues together, oxygen isotopes, carbon isotopes, sediments, and fossil assemblages, we start to see ancient worlds with surprising clarity. Reconstructing paleoenvironments is like assembling a puzzle from tiny scattered pieces. Each piece on its own might seem trivial, but together they create a landscape in which evolution unfolds. And as we move deeper into human origins, this environmental backdrop becomes essential for understanding not just how our ancestors looked, but why they became the kinds of beings they did. Let's revisit why reconstructing diet and habitat is such an important part of paleoanthropology. Fossils give us the physical structure of past organisms, but understanding how they lived, what they ate, where they foraged, and what kinds of landscapes they navigated adds depth to our interpretations of their behavior and evolution. Think about this as moving from a black and white outline to a full-color illustration. Diet and habitat fill in the ecological details that shaped evolutionary pressures. Let's look at diet reconstruction first. Teeth are some of the most informative fossils we have, not only because enamel preserves so well, but because it records clues about feeding behavior. The microscopic wear on the surface of teeth, whether it looks smooth and polished or pitted and scratched, can reflect the types of foods an animal consumed. Hard seeds, tough leaves, fibrous stems, and soft fruits all leave different signatures. Now this part is important. While microwear tells us about the foods eaten shortly before death, isotopes in tooth enamel provide a longer-term picture. Carbon isotopes distinguish between diets rich in C3 plants, such as trees and shrubs, and diets with more C4 plants, typical of tropical grasses. When we analyze enamel from fossil mammals, including early hominins, we can see whether these species inhabited more wooded environments or more open grasslands and how their diets shifted as climates changed. You might wonder how this connects to habitat. The key is that diet and environment reflect one another. Herbivores living in grassy savannas ingest the isotopic signatures of those grasslands, while animals in woodlands carry the chemical fingerprints of forest vegetation. By studying the carbon isotopes of multiple species found at a site, not just hominins, we can reconstruct the broader landscape. If many animals show strong C4 signatures, we're likely looking at an area dominated by open, grassy habitats. If C3 signatures dominate, the landscape probably included dense woodland or forest. Let's look at another tool, the fossil assemblages themselves. The types of animals present can tell us a lot about habitat. For example, the presence of grazing antelope suggests open grasslands, while climbing primates and forest-dwelling bovids point toward wooded areas. These faunal patterns, combined with isotopic data, sediment analysis, and even ancient soil chemistry, help us build a detailed picture of past ecosystems. Now, this part is especially important for understanding human evolution. Throughout the Miocene, Pliocene, and Pleistocene, Africa experienced major shifts in climate, often moving between wetter periods with more wooded environments and drier periods with expanding grasslands. These environmental fluctuations influence the availability of food resources, the distribution of predators, and the feasibility of different foraging strategies. Early hominins likely responded to these changes with shifts in diet, ranging from more fruit-based feeding to increased use of tougher, more fibrous plants, or even underground storage organs. Think about this. When we pair diet reconstruction with habitat reconstruction, we begin to see how ecological pressures may have shaped anatomical traits. Changes in tooth size, jaw strength, and even locomotion make more sense when placed in the context of shifting landscapes and food sources. A jaw adapted for powerful chewing, for instance, doesn't just reflect anatomy. It reflects a response to environmental demands. As we move forward, keep imagining fossils not just as bones and teeth, but as ecological indicators, carrying chemical and behavioral clues about the worlds our ancestors inhabited. These reconstructions help us understand not only what early hominins were, but how they lived, survived, and adapted across dynamic ancient landscapes. Let's revisit why we spent so much time examining ancient environments, isotopes, and habitat reconstruction. All of that work points us toward a central question in biological anthropology. How did environmental change shape human evolution? Evolution is not just a story of anatomical transformations, it's a story of organisms responding to shifting climates, landscapes, and ecological pressures. When the world changed, early hominins had to change with it, or risk disappearing. Let's look at the big picture. Throughout the Miocene and into the Pliocene, Africa experienced significant cooling and increasing climatic variability. Forests contracted, grasslands expanded, and rainfall patterns became less predictable. You might wonder why this matters for evolution. It matters because changing environments alter the distribution of resources, food, shelter, water, and those pressures select for traits that improve survival under new conditions. When early hominins found themselves navigating more open landscapes, for instance, behaviors that worked well in closed forests no longer offered the same advantages. Environmental instability, rather than a steady directional shift, seems to have been a major driver of hominin evolution. Studies of oxygen and carbon isotopes from sediments and fossil remains show repeated cycles of wet and dry periods. These fluctuations meant that early hominins faced not one stable environment, but a mosaic of changing habitats. In such settings, flexibility becomes a key adaptive trait. Traits like broader dietary breadth, increased mobility, and the ability to exploit different ecological niches would have been strongly favored. Think about bipedalism in this context. While no single environmental explanation fully accounts for why hominins became upright walkers, shifting habitats likely played a role. Moving efficiently across patchy landscapes, spotting predators at a distance, or freeing the hands for carrying food could all have provided selective advantages under conditions of environmental variability. Likewise, changes in dentition and jaw structure become easier to understand when we consider the dietary transitions visible in carbon isotope data. As grasslands expanded and food sources diversified, hominins may have increasingly relied on tougher or more fibrous plants, selecting for stronger chewing muscles or larger molars. Environmental reconstruction also helps clarify why certain hominin species emerged while others went extinct. When climates changed rapidly, species tightly specialized for one habitat may have struggled, while more generalist species, those capable of exploiting a wide range of foods and habitats, had better odds of surviving. This pattern appears repeatedly in the fossil record and offers insight into why our own lineage ultimately prevailed. As we pull these threads together, think about how understanding past environments enriches our understanding of human evolution. It allows us to connect anatomy with ecology, behavior with landscape, and adaptation with climate. The fossil record gives us snapshots of form. Environmental reconstruction allows us to animate those forms within the worlds they once inhabited. And as we move forward into later modules, that ecological lens will remain essential for interpreting the success, diversity, and resilience of the hominin family. Let's revisit what we explored in this section. We stepped into the ancient worlds our ancestors inhabited, using isotope chemistry, sediments, and fossil communities to piece together climates and ecosystems long vanished. These reconstructions are not side notes to evolution. They are the stage on which the entire evolutionary story unfolds. Let's look at why this matters. A fossil without environmental context is only half interpreted. When we know whether an organism lived in a closed forest, a patchy woodland, or an expanding grassland, we begin to understand the pressures that shaped its anatomy and behavior. You might wonder how something as small as an oxygen isotope ratio can reshape our understanding of human origins. The answer is that these tiny chemical clues reveal broad patterns of climate change, and those changes set the rhythm for evolutionary adaptation. Environmental instability, not stability, appears to have been a major driver of hominin evolution. The repeated shifts between wetter and drier conditions encouraged flexibility, innovation, and new ways of surviving in unpredictable landscapes. When we place fossils into these dynamic settings, the emergence of traits like bipedalism, dietary diversity, and increased mobility becomes not just plausible, but expected. As we conclude this section, keep in mind that understanding ancient environments is not about recreating scenery. It's about understanding the ecological challenges that shaped our lineage. In the next modules, we will carry these insights with us as we examine the fossil record and trace the pathways of human evolution through deep time. First, let's revisit what we set out to do in this module. We began with the fossils themselves, how they form, how taphonomic processes shape what survives, and why certain materials, like teeth, dominate the record. Then we moved into the challenge of placing those fossils in time using relative and absolute dating methods to build a coherent timeline. Finally, we explored how ancient environments can be reconstructed through isotopes, sediments, and fauna, giving us the ecological backdrop to human evolution. Let's look at why all of this matters. You might wonder why we spend so much effort studying minerals in bones, volcanic ash layers, or oxygen ratios in marine shells. The reason is simple. Without these tools, we would have no way of understanding when our ancestors lived or the worlds they inhabited. A fossil is not meaningful on its own. It becomes meaningful when we know how old it is and what kind of environment shaped it. Now, this part is important. The story of human evolution is inseparable from the story of Earth's changing landscapes. Climatic shifts, expanding grasslands, fluctuating rainfall, all of these shape the opportunities and challenges early hominins faced. By integrating fossils, chronology, and paleoenvironmental evidence, we gain a fuller, richer understanding of why evolution took the turns it did. As we move forward into the next modules, keep this integrative lens with you. Evolution isn't just about bones, it's about time, place, and the dynamic worlds that shaped every step of our lineage's journey. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.